Respiratory system. You need to know the basic function of each organ system. In a nutshell, what is the basic function of the respiratory system?
To make sure that your body gets oxygen by yourself.
Who else is involved in making sure that oxygen reaches the cells? Because the respiratory system alone can't do this. Cardiovascular system, right? They work together. So really the job of the respiratory system is to provide gas exchange across the respiratory membrane, which I drew yesterday. You have the little air sac surrounded by capillaries. The wall of the air sac is made of what type of epithelium? Simple squamous. And why simple squamous? It is the thinnest, easiest barrier to pass through, right? So the distance for diffusion is very short. Likewise, the wall of the capillary is made of simple squamous epithelium. So you take the two simple squamous epithelia, you sandwich them together, that's your respiratory membrane. Right? That's the barrier separating external environment from internal. And that allows for gas exchange. Now it's the job of the cardiovascular system to keep moving this blood. Right? So keep bringing blood back into the lungs that has high CO2 that we can then get rid of. And at the same time, you're bringing back blood that has less oxygen. So we're going to oxygenate the blood as it passes through and remove the CO2, and then it's off to the tissues again. Basic function of respiratory system.
Gas exchange.
Brain fart. Basic function of urinary system. That's what I want to say. Urinary. And there's dozens of specific functions, so this requires condensing, sort of summing it up. Regulates the volume and composition of the extracellular environment. Blood. That's the most important one, right, out of all the types of extracellular fluid, because that's the one we monitor with receptors. That's the one we make adjustments to. That's what the organ systems, 10 of the 11, are making adjustments to all the time. Volume and composition. And that encompasses a lot. Electrolyte levels, nutrient levels, waste levels, osmolarity, pH, volume, and therefore pressure. So there's a lot going on with that urinary system. Digestive, basic function. To bring nutrients, raw materials from the external environment into your internal environment. And the word digest, what does that mean? Breakdown. What are we going to use to break down those nutrients?
Enzymes.
Enzymes. What are enzymes? They are proteins, yeah. What types of reactions are these enzymes catalyzing in the gut during digestion? Catabolic, absolutely. These reactions involve water. So another way to describe them is to say they are hydrolytic reactions, hydrolysis reactions. And so all digestive enzymes are hydrolytic enzymes. They all promote hydrolysis. That's how we break down biomolecules, right, into their subunits. We can't absorb a protein or a triglyceride or a polysaccharide, but we can break them down into their component parts and absorb those. the amino acids, the monosaccharides, the fatty acids. What's the opposite of hydrolysis? Dehydration synthesis. So once you bring the monosaccharides and amino acids into the internal environment, which really means into the bloodstream, they get picked up by cells, and what do cells do with these raw materials? Two basic things, two options. The raw materials we're bringing into the internal environment from the external environment. Why are we doing it? What do we need them for?
Synthesize energy, store
Can you synthesize energy?
and break it down and use it.
No.
You extract it from the external environment, right? Energy, I'm glad you brought that up, cannot be created or destroyed, right? Only converted from one form to another. So fuel, right, is a good way to sum that up. Some of these raw materials are used as fuels, like fatty acids from our lipids, the dietary fats. Monosaccharides in the form of glucose, right, from the starch and glycogen that we consume. And when you assemble those monosaccharides and those amino acids into polysaccharides and proteins, that's all dehydration synthesis. Aside from energy, what else are we using them for? Even as adults, especially as children, growth, maintenance, repair, right? To build cells all day long. You turn your cells over. Some of them don't turn over. Some of them turn over at a very high rate. What are some examples of cells that have poor regenerative or no regenerative capability?
Cardiac.
Cardiomyocytes, neurons, and the cells that have a high turnover rate, getting replaced all the time. Epithelial cells. Well, what do you need to make a cell? What are the basic materials? Lipids,
Phosphate.
carbohydrates, proteins, nucleic acids, right? The four basic biomolecules. Those are the building blocks for making cells.
Can you repeat those, please?
I'm sure someone else can.
So we have
Four biomolecules.
lipids, carbs,
Proteins, nucleic acids.
proteins, and nucleic acids. Thank you.
What are the building blocks of nucleic acids?
Nucleotides.
Nucleotides. What are the components of a nucleotide? This feels like class. It doesn't feel like a review. You're supposed to be giving me answers. Remember, I'm not supposed to talk too much. Bad for my voice. Need a break. 3 basic parts.
Hydrogenous base,
Mhm.
sugar,
Mm-hmm.
is it phosphate?
Yep.
Okay.
It's the modern triglyceride, right?
The what? Sorry.
Modified triglyceride.
The nucleotide. We're looking at the component parts of the nucleotide. How about the sugar? More specifically? Pentose sugar. What are the 2 variations of the pentose sugar? 2 types. It's part of the name of the nucleic acid itself. So what does DNA stand for? Deoxyribonucleic acid.
It's ribose.
Deoxyribose for DNA, or for RNA, ribose. Ribonucleic acid, ribose. How about the nitrogenous bases? What are they?
Amino acids?
They're nitrogenous bases, but what's their name? How many are there? 5.
Uracil, adenine,
Uracil, adenine,
Thymine.
thymine, guanine, cytosine. Of the 5, which one is not used by DNA?
Uracil.
Which one's not used by RNA? What do you call a sequence of 3 nitrogenous bases? Triplet. What does a triplet code for? Not a protein. 1 triplet codes for 1 amino acid. So in terms of triplets, what would a gene be? A sequence of triplets, right? A bunch of triplets. If there's 200 triplets, what does that tell you about the protein? Big or small? Are we going to call it a protein or are we going to call it a peptide? We call it a peptide. What was the cutoff? If you have 200 triplets, how many amino acids do you have?
20.
200. 1 triplet codes for 1 amino acid. 200 triplets, 200 amino acids. Is that a protein or polypeptide at 200?
protein.
Protein.
You're over 50, right?
Over 100. And if it's below 50, we usually just call it a
Polypeptide?
peptide or polypeptide. Between 50 and 100, take your pick. That's the gray area. So as examples, I mentioned oxytocin, antidiuretic hormone. Those are considered peptide hormones. If you called them a protein hormone, you wouldn't be wrong. But peptide's a better term because it indicates it's really small. And each one of those hormones is only 9 amino acids in length, really small. Closer to 200 would be growth hormone, insulin. So those are always referred to as protein hormones, just based on the size. Have you thought of any questions yet? I can go all day long. But I can't cover everything in 3 hours, so I'm gonna make sure I cover what you guys need. All I hear is a fan above me. Resting membrane potential. That's a good one. What if it's all the constants I get? I feel like I used to have really good hearing. What? And again, the approach that I'm taking right now is the approach you want to take when you're studying at home, or better yet, with a study partner. And that is pick a topic and explore it. Take it every which direction you can until you've covered everything that's related to it, right? That we've actually covered in class. Don't go beyond that. So like with buffering, that was it. There really wasn't anywhere else we could go with it. We defined what it is, the 2 examples that we provided in class, the buffering system. And what is the name of that buffering system, by the way? The H2CO3, double arrow, H plus. Carbonic acid bicarbonate ion buffering system, or buffering reaction. But I usually condense that to bicarbonate. But it does have a full name. And the full name does tell you the name of the weak acid and the weak base, right? That's what the name is. Carbonic acid bicarbonate ion buffering system. Start with the basics. Resting membrane potential. Start with the name. That's always a good place. Resting. So this thing called the resting membrane potential is something we see when cells are at rest. Well, excitable cells are not always at rest. Sometimes they are excited. And if you measure the voltage across their membrane when they're excited, you get a very different value. And it actually changes very quickly. It's called an action potential. When the voltage, which has been at rest for a while, suddenly spikes up, just as quickly comes down, and then right back to where we started. And when you see this thing, that's not where we started. When you see this thing on a graph, they're referred to as spikes or action potentials. Here, the voltage is getting more positive, like plus 15. Resting membrane potential, maybe minus 70 for a neuron. And that's a little too low. Maybe more like that. And then this is like minus 80. And it's called depolarization because when we're at minus 70, the membrane's polarized. In what way? So we've used that term polar a few times now. The term itself just means what?
Unequal sharing?
That's specific to a covalent one. Just the term polar. Yeah, the thing has differences to it, right? If it's structured, opposite sides to it. All right, so the membrane, why is it polarized at rest? What is it that's opposite, different? What two sides are we looking at? So think about when I draw it, I'll indicate the voltage, capital V voltage, M membrane. A membrane potential is measured as a membrane voltage, say minus 70 millivolts. And then you'll see me do this on this side, and this on that side. I usually mark them. You could do ECF, ICF, out, in. So this is the membrane, exterior surface, interior surface. So what's polarized at the moment? Or how is the membrane polarized at the moment?
Two sides.
One side has way more positive charges, and the other side has way more negative charges. If we had equal numbers of positive and negative charges on either side of the membrane, it wouldn't be polarized. So if you go from a voltage of minus 70 to minus 60 to minus 50 to minus 40, 30, 20, minus 10, 0, you depolarize the membrane, right? Because at 0 millivolts, it's not polarized anymore. 0 means equal numbers of positive and negative charges on either side. So that upward swing of the action potential is called depolarization. And then when we come back down, repolarization. And this is stuff we'll cover in muscle phys and neurophys. What you need to know now is, it is a form of potential energy. The units are millivolts. What we're measuring is voltage. Voltage across the membrane, comparing the exterior surface to the interior surface. And it's called resting membrane potential because it's a form of potential energy. Potential energy is based on position. We've positioned a bunch of positive charges on one side, negative on the other. Positive charges, they want to go inside. Negative charges, they want to go outside, but we're not letting them just yet. So when it's time, we open channels in the membrane. And if we open a channel for sodium, that means only sodium can go through it, and it will, what direction? Sodium wants to go in, right? Because it has a much higher concentration outside. Lower inside. So why is sodium coming in? Let me explain why that's happening.
Concentration gradient.
There's a concentration gradient, and sodium is simply moving down its concentration gradient, so it's just the kinetic energy of sodium moving it across the membrane. What else is moving it across? Is it pushing it or pulling it, however you want to look at it?
Is sodium?
So sodium's an ion. It's a cation. That's important. It's got a positive charge. It's trying to go to the negative interior. It's attracted to the negative interior. So not only does it have a concentration gradient, it also has an electrical gradient. And then you'll see later, muscle, nervous system chapters, we call it the electrochemical gradient, the net force. Right? Those two things combined will determine which way the ion moves. So when sodium all of a sudden starts rushing across the membrane to the interior, that's when we see on a graph that upswing, right? Depolarization. We're going to close those sodium channels, and then we're going to open channels for potassium. So this is now a potassium channel. Which way does potassium want to go? Higher concentration inside, lower concentration outside. Now there's two forces, of course, acting on potassium because it is an ion and the membrane is charged. So based on the concentration gradient, it wants to go out, but based on the electrical gradient, it wants to go in. So if I tell you that potassium is still going to go out at minus 70 millivolts, what does that tell you about those two forces? Which one must be stronger? The concentration gradient directed out, right, must be stronger than the electrical gradient directed in. And that's why potassium continues to leak out, even though it's attracted to the negative interior. So as potassium continues to leak out, it makes the inside negative, the outside positive, and it kind of stabilizes at around minus 70 millivolts, and that's it. That's the resting membrane potential. Now, of course, that can't be all that's happening. If it was, eventually, all that potassium leaving would cause the concentration outside to go up, concentration inside to go down. Eventually, they would equal out, and potassium would no longer have a concentration gradient. But that never happens. Why?
Sodium potassium pumps.
Sodium potassium pumps constantly grab sodium that leaks in, pumps it back out, potassium that leaks out, pumps it back in. So these concentrations are very stable. That is the sole job of the sodium potassium pump. And since all cells have them, whenever you look at the ICF of any cell, high potassium, low sodium. You look at the ECF around any cell, high sodium, low potassium. So all cells have a resting membrane potential. It's always negative. The values you'll see are typically between, say, minus 50, minus 90, somewhere in there. And excitable cells use them to generate electrical signals. That's why we call them excitable cells. They're capable of becoming excited, generating electrical currents. And what are the two excitable cells that I've mentioned? They're the two main ones of the body. Muscle cells, where the electrical signal triggers contraction, and neurons, where the electrical signal triggers release of neurotransmitter. Exactly. What is neurotransmitter? It's an example of what? A chemical signal. What are some other examples of chemical signals or chemical messengers, other groups?
Hormones.
Hormones. So eicosanoids are an example of one of these groups. So it does start with a P, yeah? Paracrine factors. Paracrine factors, hormones, neurotransmitters, those are the three main groups. By definition, what's a hormone? A chemical messenger that uses the bloodstream to reach its target. Any chemical released from a neuron that travels across a synaptic cleft to reach its target is called neurotransmitter. And you can have the same chemical functioning as all three. It can be used as a paracrine factor, a hormone, a neurotransmitter. All that matters is how is it reaching its target, and that's how it gets the different name. Who's responsible then for the resting membrane potential?
Potassium ion.
And what is it doing? It is responsible, but it's what it's doing that keeps it negative. The constant
Pumping.
leaking. The leaking of potassium across the cell membrane is what keeps it negative and positive out. The pumping doesn't generate the resting membrane potential. The pumping is just to maintain the concentration gradients. The leaking of potassium out of the cell keeps it negative. So the term that means moving out or leaking out, efflux, potassium efflux is responsible for the RMP. And then as you'll see later, sodium influx is responsible for exciting the membrane, for depolarizing it, creating that electrical signal. That part you don't need to know yet. What does the sign tell you? Does it indicate anything about the strength of the voltage?
No.
Nope. The number tells you the strength. The sign tells you what the what the interior is. So negative sign, there's more negative charges along the interior than the exterior. Positive sign, swap it. I think that's it for RMPs.
Question. So potassium is always positive, right?
Just the charge?
Yeah.
Yes.
Just charge. Okay.
Yes, yes.
And you mentioned that before, like right now the way it is, it's attracted. I mean, it wants to stay inside because of the negative.
Yes.
If that changes, if the inside becomes positive and outside becomes negative, does that mean that potassium now wants to leave to go out since it's attracted to
Right.
the negative?
For two reasons now, because of the concentration gradient, which never changes, that's always directed out, thanks to the pumps. And now you're talking about an electrical gradient that's also directed out. So you would have a lot more potassium leaving.
And what decides if it's positive or negative inside?
The flux of ions across the membrane.
Is
So
it like-
it's the fact that potassium is allowed to leak out that keeps it negative inside.
Is there a certain number, like if you pass this number, you stay negative?
So I think I know where you're going with that, and we will cover that later. There's a certain voltage that creates an electrical gradient that exactly matches the concentration gradient, but they're in opposite directions. At that moment, you have an equilibrium. That ion stops moving, right? Because those two forces are moving in opposite directions, and they cancel each other out. That's called the equilibrium potential. And for potassium, that's minus 94 millivolts. So at minus 70, we're not negative enough to actually stop potassium from leaving. But if we keep making it more and more and more negative, that electrical force directed in gets stronger, stronger, stronger. And eventually it matches the concentration gradient directed out. That happens at minus 94. So you go to minus 100, now the electrical gradient is stronger than the concentration, potassium starts to come in. But you're not going to see that happen. That's something we can do experimentally with the cell. We can make the membrane whatever voltage we want with electrodes. But in the body, that's not going to happen, which is why the rule of thumb is potassium always wants to leave. Sodium always wants to enter. Next topic.
Vesicular transport.
And back there, the other one? Related, that's good. Membrane transport, and then specifically, vesicular. We could spend the rest of the time on membrane transport easily. So, what's the first thing you should do? Let's pretend like you're doing this in your little group with your study partner, maybe even at home by yourself. You're going to work on membrane transport, right? That's the topic. I would map it out, right? Just so you have something visual to work from. So you know all the pieces are there, and then you can start exploring them. So what is the first division under the topic membrane transport?
Passive and active.
That's what I would go with. I would break it up into passive versus active. And now let's just get it all out there. Passive, you can break down into
3 groups.
diffusion, sure.
Diffusion, osmosis.
And just to clarify what's moving here when we say diffusion. Solutes. Diffusion of water would be another one, but then we have a term for that, so we're not going to put diffusion of water, we're going to put osmosis. Filtration. Also, movement of water. Let's clean that up a little. All right. Active.
Primary.
Primary and secondary.
Primary, secondary, vesicular. Let's do vesicular first. Yeah.
Is filtration the same as facilitated diffusion, or is that different? It's different.
Because again, diffusion solutes, filtration water.
So the diffusion covers simple and facilitated.
Mm-hmm.
Okay.
We could even break this down. And maybe just as a little clarifying note next to simple, but lipids. Or even better, lipophilic. Which means lipid soluble. And then for facilitates for hydrophilic solutes. And then the two variations of facilitated, depending on the size of the solute.
Carrier and channel.
Uh-huh.
Yeah, I was thinking.
Channel, mediated, and carrier, mediated. Oh, where's the practice questions that show the membrane and a bunch of oh, that's right here. All right, so you have the membrane and you have the transporters. Everyone see this? Yep. Let me pass these ones back. So here we've got hydrophilic solutes. If you count them, there's more in the ECF, fewer in the ICF. So they want to go in. They need help because they're water soluble. What's helping them get in? And you have to be able to see the picture. Yeah, so what type of diffusion is this?
Facilitated.
More specifically?
Carrier.
Carrier, mediated, facilitated diffusion.
And that's because is that sodium or glucose?
We know it's not sodium,
Oh,
because
yes,
sodium
yes,
wouldn't
yes.
use a carrier.
Right.
It's small enough to go through a channel. It must be something bigger than an ion, bigger than water, right? Water's like the smallest thing that can go through a channel, bigger than water. So yeah, it could be an amino acid, it could be glucose. Sure. All you really need to know is it's too big for a channel, so it's using a carrier.
Now the carrier, is it bringing only one type in? Is it more like kind of like a
This one is.
key and lock?
Yeah. No, they all are, but some have more than one binding site.
Oh,
Right, so you have the key and lock for maybe 2 different solutes. They both have to bind, then it changes shape.
Oh, I got you.
Yeah. But they're all specific binding sites, absolutely. And then next to it, we see hollowed out transmembrane protein. And if it's a transmembrane protein, is it integral or peripheral? If you have a transmembrane protein, is it peripheral or is it integral? It would have to be, because peripheral proteins are just on the surface of the membrane, the exterior surface or the interior surface. Integral spans the width of at least one of the two layers, typically both. And if it's both, transmembrane, all the way across. It's hollowed out, it's got a little pore, and so that is a channel. And most likely what's going through it? Really just two options. Water or ion, some ions, yeah. And the next one, they show sodium getting moved out and potassium getting moved in, and it shows the little molecule of ATP next to the transporter. It is active, more specifically, primary active, directly uses ATP. So if it's directly using ATP, what does that tell you about the solutes that it's moving? All the solutes that are getting moved are moving against their gradient, all of them. In this case, just two, but it can be more than two. But what if one of them was going down their concentration gradient and the others were going against?
Secondary?
And then it would be secondary active, and you wouldn't need ATP to phosphorylate that transporter. You would have the kinetic energy of one of those solutes. And then the last one, we've got sodium going in. And does sodium want to go in?
Yes.
So it's going down its gradient. And then we have another substance that's more concentrated in than out, and it's coming in with sodium. But if it's more concentrated in than out, it's going down or against its gradient. Sodium's coming in, this other thing is coming in, sodium wants to come in, but the other thing doesn't. It's more concentrated inside. We're going against the gradient. So one's going down, one's going against. This is secondary active. As long as one solute is going against, it's active. As long as one is going down, it's secondary active. Do you have to phosphorylate this protein, this pump? Nope. Not with secondary. You're going to use the concentration gradient of that one solute going down, and that's sodium. And that's typically the case. Usually it's sodium. Based on direction, this transporter is moving sodium out and potassium in, so what would you call that? So co-transport means same direction. These guys are moving in opposite. So what terms mean that? You have some options here. Exchanger or antiporter. Mm-hmm. And if they're both going in the same direction? I'm not saying down or against, I'm just saying same direction. The transporter is moving two or more solutes in the same direction. Co-transporter or symporter. So antiporter and symporter are opposites. Under vesicular transport, what are the types?
Exocytosis, endocytosis, and transcytosis.
Is transcytosis a third type of vesicular transport?
It's both.
No. Yeah, it's just a combination of exocytosis and endocytosis.